We've been working for many years on MRI technology to map the fiber architecture of the brain. And we have been searching for principles of organization that describe how the parts fit together, or do the parts fit together. This research looked at the question of how do the pathways fit into the brain. We asked a simple question. We said, given a connection, given a pathway in the brain, what is the other stuff in its immediate three-dimensional environment? What does it pass through? If you picture the paths of the brain being like a bowl of spaghetti, you would expect nothing in particular. You would expect that its environment is complicated and arbitrary. And you would expect that each connection is a narrow, discrete object. What we discovered is that the fiber architecture of the brain is more or less as simple as you can possibly imagine. Each pathway, rather than being an isolated pathway, is a component in a three-dimensional grid. The pathways in the top of the brain are all organized like woven sheets, so that the fibers only run in two directions in the sheet and then in a third direction perpendicular to the sheet. And these sheets all stack together so that the entire connectivity of the brain follows three precisely defined directions. Now, these directions are a little bit hard to spot because in embryological life they're simple directions, but then in the adult brain they get very folded and curled up. And so that surface of the brain that you see with all of its folds has pulled the wires around with it, and so these three directions have now become greatly curved. But fundamentally, the geometry of the brain is described by a three-dimensional grid. There are a number of subsidiary things that follow from this. The axes of this grid are the same axes that the embryo has to organize itself. So they are the nose to tail axis, the left to right axis, and the front to back axis. A second point is that the fiber pathways are not really isolated structures. In a sense, every pathway in the brain has a relationship to every other pathway given by their mutual position in this single universal grid structure. A third point is that this grid structure is strongly conserved across different species. So you see the same structure we looked at half a dozen different primates, including humans, and more or less the same structure is always present, but as you go from small, simpler brains up to larger and more complicated brains, it gets more highly curved and develops certain branches and turns. But the basic theme of this structure is preserved across. So, as a result of this, we now have a way of seeing the structure of the brain as a single unified whole. It no longer seems like a bunch of uncorrelated or isolated connections. The entire connectome fits together into a single framework which expresses developmental rules, and we speculate or hypothesize functional rules as well.